Well, now that we know what the standard form of DNA looks like, at least the Watson-Crick version, is the beta form, I thought I'd talk just briefly about some alternate forms that are known from biological systems and what their uses or importance might be. Now, in the laboratory, of course, you can take DNA and make it do all sorts of things, but we're talking about some known biological forms, at least the ones that we know about at the moment. So the three that are very commonly talked about are, of course, the beta form, which is the classic Watson and Crick form. That's this center form here with about 10 bases per turn, right-handed double helix. There's also what's known as an A form, which is a more condensed form. It looks something like this. And that form is known to exist in systems. And then there's this fairly odd form known as a Z form, which is a left-handed helix. It's also found known to be existing in biological systems. So what do they have? Well, obviously, since they're there, they might likely serve a purpose. And so people have hypothesized about some different functions that may exist for each one of these. So I thought I'd just very briefly discuss those. Of course, the beta form is by far the more common biologically significant one, given it's in pH 7 or so cell systems, generally with aqueous low salt. It's the form that you'll find. If you're going to extract DNA from whatever, that's the form that Watson and Crick described, and this is the one that you certainly would use as a model for DNA for working problems and other things. And it has some general things like 10 base turns per molecule, as we said. It has the major and minor grooves. It's about 20 angstroms wide, etc. Right? So if we look at these other three forms, we see that there's this A form, and the A form is both wider, because you think about anything, you take a coil or anything, and you start to compress it a little bit, it's going to have to expand widthwise, and that's exactly what happens here. The classic beta form is about 2 nanometers in width. The more condensed form, the A form, is about 2.3 nanometers, which is a fairly significant widthing of it. The Z form on itself is much less. It's about 1.8, so it's not as wide. Okay? Once you compress it, then you also have the number of bases per turn. Remember, we're talking about this being a spirals type of function. In the A form, you have about 11 bases per turn compared to a little over 10 that we record in the Watson-Crick model, the beta form. And then in the Z form, which again is reversed, right? It's a left-handed helix. And it's a weird irregular helix. It's got 2 bases per, instead of 1 base level, it actually has 2. You notice there's always 1, 2 base setups here. So while it's still a connection of 2 bases, A to T or G to C, it's actually a 2 base stack instead of just being 1 base pair like this. It's a 2 base stack as you go up. And of course it goes the other direction, making a somewhat irregular shape to it. So these are forms. Now the beta form, again, is the most common that we see, and is certainly the one that we would expect to be found in most biological systems. So what do the others do? Well, the A form is definitely known from dehydration conditions. So things like in bacteria, where they're desiccated, they're dried down or out of water, in low water conditions, then you certainly see this compaction. It's also thought that the A form is probably important in packing and in certain bacteria, that it's a way to get a more condensed chromosome into the bacterial forms, which don't have a lot of the complex types of chromatin that we're going to see later on when we talk about eukaryotic DNA. So it's one of the ways that we can have a compression of that and get more packed in. Also, it's pretty clear that there's a transitional form between the A form to a Z form. It's been well known that the A form is usually there before the Z form appears. The Z form has a couple of ideas. One idea is that when you start to read off the code, right? So you've got double-stranded DNA. You've got to pull it apart to read it. Well, of course, you don't pull the whole chromosome apart. You just pull out the gene region that you need. Well, when you start to pull any 2 strands together they're coiled, they start to tighten up, right? Above and below each strand, you're going to have tightening. And during replication, there's specialized enzymes that come along and actually nick the DNA so that that supercoiling disappears. But in transcription, the period of when we're actually reading off the code, making message, on each end of that area, you're starting to get this sort of condensing going on. And it's thought that the DNA, what it will often do is torque into this form where you have a Z form. It'll actually flip around in the back way, go into this more elongated form, looks something like this, and that's partially what it's doing is releasing some of this tension off of the helix for the thing. So it's found associated with transcriptional areas in certain groups, and probably it's potentially that it's fairly common as a transcriptional element. Okay, so we have normal DNA, we have compressed DNA, and then we have this sort of Z backwards form DNA, and clearly there's a link between going from the A form to the Z form.